and a wonderful good evening to all of you. It is a pleasure to see all of you here. A particular pleasure when it's on a sort of cold and wintry day, particularly in the season when people are usually off to parties. It is great that you assembled here in our auditorium to listen about the depression and the immune system. Now, really, that's not why you're here. We're all here because this is a professorial lecture by Carmine Parianthes. And when I called him up and I said, Carmine, I have to introduce you, what should I say about you? You can expect what he said. He said, no, you shouldn't, don't say anything. I said, I have to tell them something. It's my job. I have to stand up. I have to welcome people. I have to tell them something about you. He says, well, you can tell them I'm Italian. I said, even a blind person knows you're Italian. That doesn't need telling. So he said, well, you can tell them that I went to school in Italy, and then I went to the Gemelli University in Rome, and then I did psychiatric training in Cagliari. I said, well, will that really be interesting? I said, what happened then? And he said, then I came to the Institute of Psychiatry. And I said, well, hang on. What does it say on your CV? You went to this Emory place and worked with Charlie Nemiroff. He said, oh, yes, 2 years I worked with Charlie in the US. And then I came, and I said, why the IOP? He said, it's the place to be, because that's where Robin Murray was. So I said, well, that's a good reason to come, but what happened to you after that? I said, can I tell people about the 200 papers that you've published, the 5,000 citations that you have? He said, no, don't tell people about that. So I said, okay, we won't talk about that at all. And I said, well, can I tell them about the NARSAT award you got from the Americans and the award you got from the British Association of Psychopharmacology? Can I tell them that you were the Academic Psychiatrist of the Year by the Royal College in 2012? He said, no. So I didn't. I didn't tell you that. I said, look, is there something I shouldn't tell them? He said, you shouldn't tell them that I make movies. And he does. So here is one man, and it is only one man, though there are two ladies here. There is only one man, Carmine Parianthes, who in the same breath can talk to you of the work that he has done understanding the molecular genetics and the molecular biology of stress. He's the man who can tell you about animal models of depression. He can simultaneously tell you about how to take care of mother-baby interactions when the mother has psychosis and the children are vulnerable. He's a man who's written books about depression and perinatal psychiatry in the 21st century, and he's a man who makes movies. There are few people who are Renaissance professors of psychiatry, and I think we have one here. Carmine, the floor is yours.
Thanks, Shitij. I'm glad that you haven't raised any expectation for tonight. Thank you for this warm welcome. I know you're back from the US this morning, so it's really nice that you're here. Thanks to all of you for being here. Shitij just said this already. It's Christmas time. You should be out there having drinks. There'll be drinks afterward anyway, so you'll get that. And I want to thank in particular those of you who are wearing Lady Gaga or Madonna t-shirts. So this just started as a joke, and then it kind of grew into an occasion to raise money for charities. So, it's a good way to add a slightly different touch to this lecture. Okay, I need to make 3 announcements before I start. First of all, there will be data. I know that you were expecting 45 minutes of stand-up comedy, but I cannot deliver this yet. So there will be some data. Second, this talk is Athena Swan compliant. There are unequal numbers of naked men and naked women in my talk. Just kidding. And the third is that the title is scientifically based. Now, I know what you cynic out there are thinking. Oh, I just mentioned Lady Gaga and Madonna so that attract more people. Catchy title, cliches. No, this is a scientific title. And I'm serious now, because what I want to do is to show how the transition from Madonna style to Lady Gaga style mimic our understanding of the relationship between depression and the immune system. In fact, the relationship between the brain and the immune system. More, there are two key moments in the history of psychoneuroimmunology which coincide with the arrival first of Madonna and then of Lady Gaga on the scene. So this was a totally scientifically based title. Kind of. There's also another reason. And obviously, for those of you who know me and my research group may understand what is the reason. We like to dance. Can I have the lights off, please? Okay. I haven't started my talk yet. There's one more thing I need to say about the title. So this very good friend of mine, Maxine Patel, that many of you know, she's not here, she's in the States tonight. She asked me, so why I dedicated my talk to Madonna and Lady Gaga, and I didn't dedicate my talk to really my intellectual icon, the man that has inspired me throughout all my life, that really has influenced my life more than anybody else. And this is, of course, as you probably know, James Bond. I couldn't really find a link between James Bond and depression and the immune system. I tried and I tried and I tried. No, at least not a clear link as I could find with Lady Gaga and Madonna, so here we go. But for those of you who are fan of James Bond, I thought I should use this moment to remind everybody this is my inaugural lecture, so this is also about my personal journey. And, you know, when I was a child, I really wanted to be James Bond. That's me. You needed the drink before coming in, okay? Come on, show me some energy. I'm the one who is suffering here, not you. Okay, the good things about becoming a professor is that, you know, at some point, you actually manage to be a secret agent. I thought I should embarrass my students since they always, you know, I always embarrass them. This is the time for. Okay, let's get serious for a second. I do want to show some data. I do want to talk about depression and the immune system. It's the field of the relationship between the brain and the immune system is really, really exploding at the moment. And it's very important that, you know, for our understanding, our new understanding of the biological basis of mental disorder. Now, we all know what depression is, this clinical condition, persistent sadness, a lack of hope, lack of motivation. We also know what the immune system is. You know, this biological system of cells that allow us to stop virus and bacteria, but also protect us from internal trauma. But more importantly, it's a system that can influence our behavior. Now, the idea that there is a communication, a bidirectional communication between the brain and the immune system, today is part of common knowledge. We always say, oh, I'm stressed, that's why I got this infection. I'm tired, I'm working too much, and that's why I'm ill. We understand easily that there is a relationship. But this was not the case 30 years ago. This is a very new concept. And there's another point that I will make throughout my talk, which is the fact that at the beginning, the relationship between the brain and the immune system was really hierarchical with the brain on top. It was all about the brain influencing the immune system. But actually, over the years, and certainly over the last few years, there's been a switch, a completely shift in the change in our understanding, with the immune system really becoming the most important, or one of the most important biological systems in the body, controlling or influencing brain function, and potentially a system that we can target to understand, you know, maybe to offer even the drugs for the future. So, this psychoneuroimmunology starts in 1982. And you probably remember who else starts in 1982. Can I have the lights again, please? Isn't she great? I mean, I don't remember. I wasn't born in 1982. But some of you must know this. Some of you were dancing in 1982. So you must have really known, you know, Madonna when she first came out. I didn't. But in 1982, a really important paper came out. The title is obviously technical and boring, behavioral conditioning suppression, but was really revolutionary in our understanding of the relationship between the brain and the immune system. So let me step back for a second. This is our understanding of what conditioning is. Everybody knows this. So you ring a bell, and a dog doesn't notice. You ring a bell, you give the dog food, the dog starts salivating. You do that long enough and repeat it enough, and then you ring the bell, and the dog salivates even without food. That's simple conditioning. And you can understand this from the brain point of view. Brain stimuli, brain reaction. But what these papers show is that you can condition the immune system through a brain stimulus. And I think that was really revolutionary. So they gave saccharin to a rat, and obviously the immune system didn't change. Saccharin is just a sweetener, so it's a sweet stimuli. Then they gave saccharin and cyclophosphamide. That's an immunosuppressant. And obviously the rat had an immunosuppression, as you would expect, because of the medication. You did long enough, and then you give saccharin alone, and the rats respond with immunosuppression. Just showing this profound communication between the brain and the immune system that really, at that time, they could not understand the mechanism. So, but you can see how the model is clearly hierarchical, the brain governing the immune system. And over the next 10 years or so, there were at least 30 or 40 studies trying to understand the relationship between stress and the immune system and depression and the immune system. So we arrived to 1991. Can I have the lights off again? A truly Athena-compliant video. I hope you all agree with that. Nobody's been disappointed. So in 1991, well, I remember this. In 1991, I just became a medical doctor. Yes, I know, I wasn't born in 1982, and I was a doctor in 1991. I just worked really, really hard. And in Italy, you have to do an MD every time, when you become a medical doctor. And my MD was on alterazione del sistema immunitario in un campione di pazienti depressi anziani. So abnormal immune function in older depressed patients. So you can see I always loved this field, right from the beginning. So, and I managed to publish this data in an obscure Italian journal called Pan Minerva Medica, but it's in PubMed, and it's right there, my first publication in PubMed, dated 1991. I was 12 at that time. And as Shitij said, I really wanted to work in this field, and I went to what was then and still is now one of the most important laboratories in psychoneuroimmunology, which is the Andy Miller Laboratory at Emory University, where Charlie Namaroff works. And so here I am, 1995, with Andy Miller in wonderful Atlanta. And we did quite a lot of work. I worked predominantly in the lab, and there's one paper that I want to mention, published a couple of years after I left in 1999. So again, the title is technical, but that's about the effect of a cytokines, so the effect of a substance that activates the immune system on the glucocorticoreceptor, which are the receptor for our stress hormones. Okay. A picture always works to get your data published, so if you can, try to put one in. It definitely worked for me. So I'll explain very simple. The details are the details obviously are not important. It's the concept that is important. So these cells have this receptor, which is called the glucocorticoreceptor, which is a receptor for stress hormones. So this receptor participates to the regulation of stress hormones. Is in effect a receptor that is in the brain. You can look at it in cells. You can mark it with a nice dye, and you can see how it tends to stain cells on the outside, not on the inside. Now, if you then activate the receptor by giving stress hormones to the cells, the receptor nicely moves from the outside to the inside of the cells. Can you see how the center of the cells now is all very bright? But if you then treat cells with an inflammatory signal with interleukin-1 and then put the stress hormones on top, the receptor doesn't work anymore. So here you can see the nucleus remains dark, even if they got the stimulation with the stress hormones. Now, why is this important? Well, up to that point, as I've said, everything was about the brain communicating with the immune system through the HP axis and glucocorticoid hormones, through sympathetic nervous system, parasympathetic nervous system. There was only a small arrow going up from the immune system up to the brain through cytokines. And these are receptors that are in the brain that regulate the stress response. So this paper was one of the first papers showing that immune system regulate brain function, regulate stress function, as much as stress regulate the immune system. It really allows to start changing our understanding of the bidirectional communication. The hierarchical system is not, of course, changed yet, but it starts to wobble a little bit. People try to look at this from different point of view. Okay, 10 more years are gone. We're in 2001. Can I have the lights off again? Well, I didn't choose the actors in the movie, okay? So I cannot I cannot get any blame for the driver. But the beginning of the 20s was really very exciting because a new models start to be developed to try to understand the relationship between the brain and the immune system. And this models is studying patients with viral hepatitis. That you can see is an inflamed liver. Viral hepatitis, because the treatment of viral hepatitis is interferon alpha. So you stimulate the immune function through a powerful immune activation to treat hepatitis B and hepatitis C. And what's interesting about this treatment is that about a third of patients develop significant depression. About 50% develop some symptoms. About a third develop clinically significant depression. So it's a tool to see how does increased immune function leads to depression, if you like, how the immune system regulates the brain rather than the other way around. And we started doing at that time quite a lot of work. So these are data from a British court in which you can see on the x-axis the time, because treatment with interferon alpha lasts up to 1 year, so 50 weeks. And on the y-axis, you can see depressive symptoms. You can just see depressive symptoms going up and up and up throughout the treatment. And we've now replicated this in 3 different cohorts, including an ongoing studies. So this is about immune function making you feel depressed. But the opposite communication is also active. So we published in 2005 in Molecular Psychiatry that patients that develop more depression and more fatigue during interferon alpha are also likely to show less virological response. So the interferon alpha doesn't work. So immune activation leads to depression, but depression leads to abnormal immune function so that the virus is not cleared. Again, I just want to stress how this is just kind of wobbling the principle of a hierarchical structure and just allow us this communication between the brain and the immune system is really becoming more and more egalitarian, if you like. But it's really in 2008 that another papers arrived which changed the field, or at least put finally together in writings, spell it out what many people, including us, have already been saying for some time. And you also know who else arrived in 2008, right? Can I have the lights off? Well, when I saw this, I was just blown away. Seriously, I mean, people make fun of me because I'm such a fan of Lady Gaga, but I truly really like her. I mean, I think just a revolutionary artist. I can talk about Lady Gaga for the next half an hour, but I won't. But you know, she was writing songs when she was 4. Okay? Hello, PhD students here. Okay? And she didn't desist. She was kicked out of the first music school that she went. Her first agent interrupted a contract with her because he didn't trust her. Okay? So persistence. Okay? Junior academics, persistence. So in 2008, this paper came out from Robert Dancer. He is really one of the world leaders in research on depression and inflammation. And he just spelled it out. He just spelled it out the way that all of us wanted to say. From inflammation to sickness and depression, when the immune system subjugates the brain, the hierarchical shift could not be stronger. I'll just read the abstract. I think it's so clear. In response to a peripheral infection, innate immune cells produce a pro-inflammatory cytokines that act on the brain to cause sickness behavior. Now, sickness behavior is a technical term. Actually, it just means the symptoms that we all experience when we have a bad flu. Lack of energy, we just don't want to leave the bed, we don't want to go out, we're irritable, we don't have any appetite. This is just a physiological reaction to inflammation that the body activates to allow us to conserve energy and fight the infection. But you can also see how similar some of the symptoms are to the symptoms of depression. So the abstract continue. When activation of the peripheral immune system continues unabated, such as during systemic infections, cancer, or autoimmune diseases, the ensuing immune signaling to the brain can lead to exacerbation of sickness and the development of symptoms of depression in vulnerable individuals. So that's the strength of this paper. For the first time, he really said, it's not about the brain influencing the immune system. Yeah, maybe there's a little bit of effect, but it's the immune system that can really influence the brain and can really make you depressed. And how? Well, once you've established this hierarchical system with the immune system controlling, subjugating the brain, what is the mechanism? Well, he described two mechanisms, again, coming up from studies that have gone on for over decades by then. In the top, you can see our vagus nerve. These are nerves that controls our peripheral function, the heart, the lungs, the bowels, but they also collect information and sensory perception from our body. And they also collect information from inflammation. So if there is peripheral inflammation, these nerve fibers get activated. And they transmit the signals back to the brain, and specifically in areas which are in the top here delineated, that are important to emotional control, like the bad nucleus or stria terminalis. So that's a direct action of the immune activation in the periphery regulating behavior. And also, as you can see in the bottom figures here, the area of the brain where there is no blood-brain barrier. So here, the cytokines can just go. The immune signal can move from the blood to the brain and again communicate to the brain that there is immune activation. And that's enough to induce depressive symptoms. Okay? But we wanted to know more. We wanted to know how does the inflammation affect brain structure. And we started to work on the hippocampus. We wanted to understand whether inflammation could change the structure of the hippocampus, which is one of the brain areas that, as you all know, is most important for emotional control. So just a couple of years later, we published this paper, Infraceptual Psychosis, so papers, and not in depression, but still a group of people with mental health problems and high level of stress. And very simply, we demonstrated that high level of inflammation is associated with smaller hippocampal volume. So here you can see high level of interleukin-6 in the blood, and size of the hippocampus here on the left, and this nice negative correlation. So the more inflammation, the smaller is your brain. So this may be one of the mechanisms by which high inflammation leads to change in behavior and depressive symptoms, or in this case, psychotic symptoms. But just looking at the structure of something is not enough. We wanted to look at the cellular levels. So we then worked on neurogenesis. We used our famous depression in a dish model. These are cells, these are human neurons that grow into a dish, and then you can treat and model depression. And one thing we could do is create inflammatory status in a dish. And if you do that, you have less neurogenesis. You have less new neurons produced. And again, we published this in 2012. Data is very simple. You can measure neurons using antibody. You can mark them with double coating in red, MAP2 in green. These are for immature neurons. These are for mature neurons. And if you treat cells with interleukin-1, so if you increase immune function, you have less cells, 35 to 45% less new neurons. So clearly showing up to the microscopic level how inflammation affects the brain. Okay, it's 2013 now, and Lady Gaga's last CD just came out 2 weeks ago. Okay, so it's 2013, and I don't want to leave you with some unpublished data. I just want to tell you where we are going, what's the direction of our work. So, first of all, we can predict the likelihood of treatment or response to antidepressant treatment by measuring immune biomarkers. So this is a very simple scheme. You measure interleukin-1 and MIF, macrophage inhibiting factor. And based on the levels, you can decide whether the patient are green, so they will respond to antidepressant, They are red, they will not respond to antidepressant, or they are orange, they have 50% of chance to respond. So biomarkers of the immune system regulating, predicting treatment response. We are looking at the effect of interferon alpha on mRNA levels, which means the possibility of looking at hundreds of biological changes occurring at the same time in the body. And one thing I want you to notice, just by looking at numbers. Now remember, these are patients with hepatitis C. They all take interferon alpha. They all take the same treatment. Some of them develop depression, and in their body, 442 genes change. Some of them take exactly the same interferon. They don't develop depression, and only 46 gene change. So what I think this is showing is that to develop depression, you need to be vulnerable, biologically vulnerable to an immune challenge. That will make you depressed. And finally, babies. So when does this relationship between depression and the immune system start? Does it start already in utero? Our moms who are depressed in pregnancy already have high level of inflammation? Well, the answer is yes. So that's a study we are conducting at the moment. 2 key cytokines, interleukin alpha, interleukin 1 and 6, higher level in mothers who are depressed compared to controlled mothers who are controlled pregnant women who are not depressed. What we also wanted to know, what happens to the offspring of these women? What happens to the babies that were in utero when the mom was depressed? And so we have been able to follow up a longitudinal study that started about 25 years ago now, on which we have measures across the lifespan, up to young adulthood, up to about 25. And here's the data. So this is now inflammation in the offspring, immune activation in the offspring. And as you can see, offspring of mothers who were depressed in pregnancy have a high level of inflammation in adulthood, even if they are not depressed currently. This is just a signal dating back from uterus compared to offspring of mothers who were not depressed. And I really hope that with this quick summary of 30 years of research, I have to give you an understanding how important it is, this field, for psychiatry in general. And how we really need to change our understanding of the system and think about how the immune system may well become what most psychiatrists will need to understand in the future. Now, I know you want to go for drinks, but this is my inaugural lecture, so I need to thank a few people. My wife has been telling me for the last few days, this is not the Oscar. Keep it short, keep it simple. Well, sorry. This is my Oscar. Well, unless I do win an Oscar. So I do want to thank a few people. And I'll start, obviously, by thanking my wife. You can all recognize her. The serious look is the same. The inquisitive smile, the long legs. Thank you, Paula. Without you, I wouldn't be here. Then, I'm going to thank 3 people that helped me in my career here at the Institute. That's, as you can recognize them, Dinesh Bugra, Robin Murray, and Simon Wesley. Dinesh was the first person that I met when I arrived at the Institute. And although we never worked together, has always been, the brother that gave me advice at the time that I needed. So, Dinesh, thank you. Robin, my head of department for many years, and the person who was there in my transition from junior to senior academics. This is the most difficult time, as you all know. It's when it's difficult to get funding, it's difficult to get position, and it's easy to just decide to give up. And he never let me give up. So I'm grateful to Robin for this. And Simon, obviously, my current head of department that has guided me through until the professorship. Now, I like this picture. They show exactly who these people are. Robin is a serious scientist. Dinesh is a Bollywood actor. And Simon is Barack Obama. I mean, sorry, did I mention presidential election, Simon? Okay, I'm getting toward the end, don't worry. So I want to thank my group and my researchers. So first of all, I think it's just been a wonderful, wonderful journey. And only a few years ago, my research group could sit comfortably around the table. There was less than 10 years ago. And now we needed a whole stairs to accommodate everybody. So before I thank my research group, I want near me the three people that have helped me most. I want Valeria, Patricia, and Sue next to me while I'm thanking you. Come on, come on, come on. Don't start crying. So when you're a professor, or even before, they try to teach you that you should be really good in picking up people, choosing who is the right researcher, who will have a career. I was lucky because I didn't have to pick up good people. The good people picked up me. They wanted to work with me when they didn't even know me. So I'm grateful for this. And now I want all of my researchers to stand up. Come on, stand up, stand up, stand up. Come on, don't be shy. Turn around, face the audience, face the audience. Yeah, it's not fair that they do all the work and I get the credit. So this time, the applause is not for me, it's for them. Thank you very much. And we now need some technical help.
It needs to go onto the PC, not on. It's already on the PC. Okay.
Yeah, keep it going.
I should like to propose a vote of thanks to Professor Parianthes for sharing with us tonight his love of science, his love of music, and his love of life. Carmine took on the mantle of head of perinatal psychiatry in 2008, succeeding the late Professor Chani Kumar. This was some mantle. It had become apparent, not least to Professor Sir Robin Murray, who spoke to me in the lift, that the job Chani did could not be done by one single person alone. Oh, sorry, I'm now on the wrong machine. Flips into machine to press. Oh, okay. Sorry. It's a picture of Chani Kumar on the mother and baby unit. And sadly, of course, he died in the year 2000. So Carmine took on the mantle, but not alone. Indeed, it needed 4 consultant psychiatrists to take on the mantle. Carmine, a biological psychiatrist, as head of the academic section of perinatal psychiatry at the IOP. Dr. Trudis Nevaratne, as head of perinatal psychiatry services and consultant psychiatrist on the mother and baby unit. Professor Louise Howard, who took up a chair in women's mental health, and Dr. Paula Dassan as a consultant psychiatrist for perinatal services in the community. So what Chani had done needed 4 consultants. Perinatal psychiatry is perhaps one of the most difficult specialties. not least because it involves the mother, the fetus, the neonate, and the infant. It's the bridge between adult and child psychiatry. And I read in The Observer on Sunday that lots of people don't like working with babies because it's super difficult. With adults, you could just ask them questions. With animals, you can make them do things. Not so with babies, says UCL researcher Zita Patai. My 4 colleagues and I have all been lucky enough to work with Chani Kumar and have thus inherited his passion for keeping the baby in mind. Kamini has upheld all that Chani held dear. His total belief in the importance of combining clinical work with research and research with clinical work. To this end, Kamini chaired the committee that organized a perinatal psychiatry conference in 2011 here at the Institute, from which a book is currently being published by the Oxford University Press. Kamini has also contributed to the government's cross-party manifesto for the next election, the 1001 critical days, the importance of the conception to 2-year period. By combining his role as head of the SPY Lab, Stress, Psychiatry and Immunology Laboratory, of course he really wanted to be James Bond, with the section of perinatal psychiatry, Kamini is carrying out his vision of understanding the relationship between physical and mental health for the enhancement of both. Blending passion and expertise. Whittards, apparently, have been doing it since 1886 in their desire to produce the best coffee and the best tea. Kamini's passion and expertise led him to complete his doctorate in 2003. Now I'm telling you the truth here, not what he said. And since then, he has used them to enthuse his students, so that in 10 years, 11 of his students have successfully been awarded their doctorates, the 11th being yesterday. This is the rate of 1 a year, over 1 a year. Not only does his passion and expertise encourage his students to work, but also to play. And he gave you many examples of our play at the beginning of his talk. And I should have you just notice that I was not present in any of those videos. After a certain age, you can bail out after the dinner while the others go dancing. But we drink plenty of champagne. Kamini would be the first to acknowledge that his elevation to his professorial position has been made possible because of the dedication and passion that he elicits from his band of 29 researchers and students. "29, coming. You will not perhaps be surprised that during our recent SPY Lab professional development meeting, looking at the Myers-Briggs type indicators, Carmine's 4 letters were E, extroversion, N, intuition, F, feeling, and J, judgment. The mentor, represented by Apu. He's a smooth talker, charismatic, compassionate, possibilities for people, ignores the unpleasant, idealistic, most persuasive. What is perhaps even more interesting is the high number of his team who are introversion, S, sensing, F, feeling, J, judgment. Individuals with a high sense of duty who are most loyal, represented, of course, by Marge Simpson. Carmine, thank you from us all, and especially from your perinatal and spine lab colleagues and students.
So there you have it, ladies and gentlemen. We know that the Italian heritage gave us Luis Veronica Ciccone, that's Madonna, and then it gave us Stefani Germanotta, that's Lady Gaga, and there you have it, Carmine Parianthes. So thank you all for joining us today. To the IOP community, Carmine is very dear, and you can see the reasons why. You can see if while you were a student here, he'd be a mentor you would seek. You could see how if you were a colleague here, this is a colleague that you would seek. And you can see why you were a dean here, you would very much want Carmine to be here. So for all those reasons, Carmine, thank you for what you've done here. Thank you for what, by being here, you have made IOPP and all that you stand for your students and your researchers. And now, to celebrate that, let's all come to the floor upstairs, and there is wine and cheer. Thank you very much for your attendance.